We are pushing through the interior of a standard eukaryotic cell, navigating a dense, highly specialized labyrinth of folded membranes, mitochondria, and transport vesicles. But if we break through the cellular boundary and enter the microscopic space beyond, we find a completely different architectural standard, a stark, empty void. This minimalistic space is the domain of Kingdom Monera. The organisms classified here are unicellular prokaryotes, meaning they operate strictly as single, self-contained units. Biologically, they are defined by a negative trait, the absolute lack of internal subdivision. There is no true enclosed nucleus to house genetic data, and there are no membrane-bound organelles. The specialized machinery we expect in a cell is simply missing. Yet these stripped-down organisms drive the global ecosystem. Operating primarily as decomposers, they break down dead organic matter and recycle essential nutrients back into the soil and atmosphere. This presents an engineering problem. A system must store data, manufacture proteins, process energy, and reproduce. We have to understand how a cell accomplishes all of this and dominates global biomes without the complex internal infrastructure found in plants or animals. To see how they succeed, we have to drop our eukaryotic biases. We need to look at the Moneran cell as a highly efficient machine and engineer its architecture from first principles, component by component. Our blueprint starts with the bare physical dimensions of the cell, a blank, three-dimensional void waiting for instructions. Watch as the source code materializes. This genetic material is naked, double-stranded, and looped into a single circular strand. Notice this highlighted boundary. Without a membrane, the DNA floats openly in a decentralized zone called the nucleoid. In a eukaryotic cell, the nucleus acts as a heavily guarded vault, requiring complex transport mechanisms to move data in and out. Here, the open floor plan arrangement means the genetic code is continuously bathed in the cellular fluid, allowing direct, immediate chemical interaction with the rest of the cell. By stripping away the heavy physical protection of a nuclear envelope, the monerans cell sacrifices structural isolation to achieve extreme operational speed. Relying on a single primary strand of circular DNA poses an evolutionary risk. If an environment rapidly becomes toxic, a static, unchangeable genome leaves the organism with no defense. The biological solution appears here, as independent secondary rings of DNA emerge. These highlighted structures are plasmids. They contain highly advantageous genes, entirely separate from the core nucleoid data. Plasmids operate independently. The cell can replicate them, drop them entirely, or swap them directly with neighboring bacteria through physical contact. If a new threat like an antibiotic is introduced to an environment, a resistance gene on a plasmid can be rapidly shared across the population. They function as modular software updates, adapting the cell without requiring a rewrite of the core operational code. Information in the nucleoid and plasmids must be translated into physical structures. The cell requires manufacturing machinery to read the genetic code and build actionable proteins. In this animation, hundreds of granular structures latch on to the DNA. These are ribosomes. A scale comparison highlights another reduction. Monerans utilize 70S ribosomes, which are measurably smaller and less massive than the eukaryotic 80S type. Despite their reduced mass, these ribosomes process amino acids at high speeds, reading messenger RNA directly as it peels off the DNA strand. The 70S ribosome proves that metabolic efficiency does not scale linearly with physical size. Immense structural scale is not required to maintain baseline biological manufacturing. At this stage of the blueprint, the internal components are completely exposed. Without a physical boundary, external osmotic pressure would cause the cell to either rupture or collapse in on itself.
To protect the internal void, a rigid cell wall forms. This lattice-like grid tightly encloses the cell. Pulling in close on the structural formula, we see it is built from peptidoglycan, a tough polymer distinct from plant cellulose. There are strict exceptions, like mycoplasma, which survive without a peptidoglycan wall using stable membranes. Returning to our blueprint, external hardware attaches to the wall. Whip-like flagella lock into place for motility, while shorter pili emerge to anchor the cell. The addition of the peptidoglycan wall and its external appendages takes a vulnerable, naked chemical soup and secures it inside a heavily armored, highly mobile biological vehicle. Fully constructed, this vehicle must now fuel itself. Monerans draw power from diverse nutritional modes. Some are autotrophs, synthesizing food via photosynthesis or chemosynthesis, while others operate as heterotrophs, surviving as parasites or symbionts, or saprophytes feeding on decay. Once fueled, the system multiplies. Watch their rapid reproductive cycle. The cell elongates, pinches at the center, and violently snaps into two identical organisms. This asexual process is called binary fission. It bypasses complex multi-stage eukaryotic division, allowing populations to double in a matter of minutes. While binary fission produces identical clones, the population still achieves genetic diversity. They utilize modular plasmids for genetic recombination through conjugation, direct transformation, or viral transduction. By pairing the sheer numerical output of binary fission with the constant active swapping of plasmid data, the moneran architecture creates a self-replicating engine optimized for rapid evolution. To see the limits of this theoretical blueprint, we must observe its historical deployment. Biologists traditionally divide Kingdom Monera into three dominant groups, each testing the architecture in different environments. We start in the most hostile environments on Earth: boiling volcanic hot springs, hypersaline lakes, and toxic marshy zones where temperatures and chemical stressors prevent complex life from taking hold. These environments are populated by archaea bacteria. This primitive, highly specialized group uses the stripped-down moneran model, relying on unique membrane lipids to maintain stability where standard cells dissolve. Archaea bacteria demonstrate that the basic prokaryotic model provides absolute physical durability, surviving chemical extremes that instantly destroy eukaryotic tissue. Outside of extreme chemical stressors, survival depends on capturing ambient energy and processing the surrounding atmosphere. This function belongs to a second group, cyanobacteria. Historically referred to as blue-green algae, they are not plants, but fully functional prokaryotes. They utilize an autotrophic model, drawing in sunlight to perform large-scale photosynthesis. Simultaneously, many species fix atmospheric nitrogen, converting inert gas into biological compounds usable by the broader food web. By oxygenating the early Earth and continuously cycling nitrogen, cyanobacteria prove that a single cell can re-engineer a planet's atmosphere entirely without specialized membrane-bound chloroplasts. The final historical division is the eubacteria. These are the true bacteria, encompassing universal standards like Escherichia coli, and they inhabit nearly every moderate environment, host organism, and surface on the planet. Across archaeobacteria, cyanobacteria, and eubacteria, the thesis of Kingdom Monera remains consistent. They achieve global saturation not through internal complexity, but through architectural simplicity. Stripping the cell down to naked circular DNA, efficient 70S ribosomes, and a rugged peptidoglycan wall removes metabolic overhead required to maintain a nucleus and internal organelles. True biological supremacy on Earth belongs to the organisms that perfected the engineering art of doing more with less.